if you look at the last 20 years, there have been 3 coronaviral outbreaks: SARS, MERS, and COVID-19. And it's only a matter of time before another coronavirus comes knocking at our door. And so, in order to be ready for it, we want to have a lot of tools to actually address these pandemics. Over the last several years, we've been developing Nanopore DNA sequencing, but we've also been using the Nanopore as a way of understanding motor enzymes, things like helicases and polymerases. This is what the data sort of looks like. There's current as a function of time. And if you read a bunch of these, if you take 20 or 30 of them and stack them up on top of each other, it turns out that the current values that you read are really reproducible. What's interesting, though, is this trace here. You can use it to sequence DNA, but it's also a single molecule record of this motor enzyme's motion as it goes along the DNA. When we're doing this, we're measuring the enzyme's position as a function of the DNA bases. So we get a sequence-specific measurement of where the motor enzyme is along the strand as a function of time. You can really learn a lot about the kinetics of one of these motors. But what you can do is you can take ion current, and if you know what this sort of smooth pattern is, because if you know what the DNA sequence is that you're walking on, then you can convert ion current as a function of time into DNA position as a function of time. And it turns out you're able to really resolve these steps with really high spatiotemporal resolution. You can run it with one enzyme and you can look at the current levels that you see, and you could run it with a different enzyme and you can see that you see the same sort of current levels. Now, it turns out in this helicase, it's taking sort of twice as many steps along the DNA, but still follows the exact same pattern. Sort of what that means is that just by looking at where you are on the pattern of the DNA, you can measure your position along the DNA. So how far you are along the strand as a function of time. If you've got one of these graphs that has position on this axis and time on this axis, really one thing you want to know is, like, for a given step dwell time and step size, can you see it? And so this graph has sort of what's resolvable for a bunch of different single molecule techniques. Most of the motor enzymes that walk on DNA are actually faster than that. So they take single nucleotide steps on millisecond timescales. And so it's actually really hard to observe them using sort of the standard single molecule techniques. When you're looking at our data, you can observe things that are 1,000 times faster than what you'd be able to observe with optical tweezers or magnetic tweezers or something like that. Here's an example optical tweezers trace. They're looking at RNA polymerase, and they were able to see single nucleotide steps in there. Now, here's some example data from SPRNT Nanopore tweezers. If you look at the x-axes here, they're pretty wildly different. This section of data right here fits right here on this graph. And so really, you're just able to observe much faster steps with so much better resolution. And that allows you to study a whole new class of enzymes that just couldn't have been studied before. It turns out that COVID, the virus, actually encodes for its own polymerase and its own helicase. The polymerase is responsible for replicating the viral genome, and the helicase is responsible for sort of wrangling the long RNA. COVID has a very long genome, and it needs a helicase to kind of help keep it sorted out well. So if you get rid of the polymerase or you get rid of the helicase, COVID just doesn't work. NSP13 is really highly conserved among all of these viruses. And so if you have something that works against that NSP13, this helicase from COVID, it's probably going to work for SARS-3. And so that's sort of the motivation for doing this and learning more about this motor enzyme. One thing that we found that really helps get these sort of things to work is using them on the backwards pore geometry. The normal way to do it is have the vestibule upward, but you can also set this thing up with the vestibule facing downward, and the constriction's just right there, and it can capture the DNA right away. We found that it was a lot easier to get data quickly with the backwards pore, and so we decided to go with that. The helicase, even though it's an RNA helicase, it actually works equally well with DNA, and so we decided to do this experiment with DNA to really give ourselves the best chance of it working.
So every nanopore experiment begins with the enzyme NSP13 bound to either single-stranded or double-stranded DNA. It is electrostatically drawn through the pore. In this case, we're using the backwards pore geometry. The current transitions correspond to single nucleotide translocations, And we find that the enzyme runs at almost 2,000 nucleotides per second. SPRNT is the only tool that we're aware of that can measure at this really high spatiotemporal resolution of 0.5 nanometer at less than a millisecond. And next, we wanted to study the double-stranded unwinding by NSP13, and we find that similar to single-stranded translocation, double-stranded unwinding also occurs in single nucleotide steps. When we compare the average speeds of thousands of events, double-stranded DNA unwinding and single-stranded DNA translocation, we find that the speeds are 8-fold different from each other, and the double-stranded unwinding speed is comparable to the SARS-CoV-2 RTC in bulk experiments. Finally, we look at the amount of time NSP13 spends at each nucleotide position, and we find that that time varies with the sequence identity in the enzyme for both the single-stranded DNA and the double-stranded DNA. Next, we wanted to explore what happens at the single nucleotide level. The Inchworm model describes the series of conformational changes that occurs in a single ATPase cycle that leads to single nucleotide translocations. And in this mechanism, the Walker domains associate with the nucleic acid and also house the ATP binding site. Initially, they are apart. Upon ATP binding, the Walker 2A closes in towards 1A in the 3' direction, lowering the nucleotide by 1 nucleotide in the constriction, which results in a single nucleotide transition in our SPRNT data. Next, upon ATP hydrolysis, Walker 1A moves forward by a single base towards the 3' direction and completes the single ATPase cycle. Since each step transition is an individual ATPase cycle, we can place all of these series of chemical and physical steps in the context of a single nucleotide transition in our SPRNT data. This model is further validated by our ability to manipulate each of the parameters that affect the different rate constants by varying the concentration of ATP, ADP, presence of a duplex, etc. Next, we wanted to explore if we could observe the exact steps in which ATP gamma S inhibits the ATPase cycle of NSP13. We chose the inhibitor ATP gamma S. Helicases typically don't hydrolyze ATP gamma S or do so incredibly slowly. We compared the translocation and unwinding of DNA in the presence of ATP gamma S by itself and mixed with ATP. We find that in the case of double-stranded DNA unwinding, ATP gamma S slows down NSP13. Similarly, in the case of single-stranded DNA translocation, ATP gamma S hydrolysis is a lot slower. We also find that in the case of mixing 300 micromolar ATP gamma S and 600 micromolar ATP and comparing it to the condition of just ATP gamma S, we find that it saturates at 300 micromolar ATP gamma S. And when we look at the individual dwell time distributions at each position, we find that the sum of how long it takes NSP13 to hydrolyze ATP gamma S and how long it takes NSP13 to walk on double-stranded DNA matches well with how long it takes NSP13 to do both. Next, we can easily measure when the enzyme is at the start of each of these events. About 50% of the time, we capture it at the walking at the beginning of the design start site, and the probability increases when it is unwinding double-stranded DNA, and we capture it at the beginning almost 100% of the time when it is in the presence of ATP gamma S. This leads us to believe that in the bulk solution above the nanopore, the enzyme does not hydrolyze ATP gamma S, and that ATP gamma S completely inhibits ATP hydrolysis and duplex unwinding. However, when supplemented with an external force from the nanopore, the closed conformation is favored and hydrolysis occurs, even though it occurs slowly.
Of course, ATP gamma S is not a viable drug candidate or anything. But what's cool here is if you can really understand mechanism, you can really start to improve mechanism and go through and make better inhibitors.
Theoretically, if you were to design an inhibitor to interfere in any of these sub-steps, SPRNT can measure the rate constants and how effectively you inhibit any of these steps. And that is further information for how to optimize to enhance inhibition, for example.